everyone, and welcome to another episode of GenLive. On today's show, we'll be discussing spatial biology and spatial omics. I'm Julianna Lemure, science writer at Gen, and I'll be your host today. I'm joined by two of my colleagues and GenLive regulars, Alex Philippides and Kevin Davies. Hi, Alex. Hi, Kevin.
Hello, Julianna.
Julianna.
Hi, everyone.
Hi, Kevin.
Before I introduce our guests and get our conversation started, I want to mention that this episode is GenLive's 1-year anniversary show. If you remember, we started the show last year with Alex and I as the guests, talking about what we were learning about COVID-19 at that time. We've come a long way since then, and I don't know about you, but I've really enjoyed doing these shows every month. So happy anniversary to us and happy anniversary to GenLive.
Thanks, Julianna. Yeah, happy anniversary to us.
First, a quick word from our sponsor.
Yeah, this episode of GenLive is brought to you by GenEdge. GenEdge is the new premium subscription channel from genetic engineering and biotechnology news, providing in-depth, exclusive reporting on the biotech industry from the bench to the boardroom. GenEdge provides detailed profiles of innovative and disruptive biotech companies, CEO interviews, analysis of major funding rounds, an exclusive multimedia series, including Close to the Edge. You can test the waters with a free 30-day trial, www.genedgenews.com/GenEdge. Julianna, over to you.
Thanks, Kevin. So I would like to introduce our guests. We're joined by three very special people today. Josh Rue is the co-founder and chief technology officer of Rebus Biosystems, a spatial transcriptomics company located in Santa Clara, California. Josh was one of the original developers of the company's core technology, synthetic aperture optics, or as we'll say, SAO, during his graduate work at MIT. Today, Josh will tell us more about SAO and how spatial transcriptomics is disrupting the world of genomics. Hi, Josh. Thanks for joining us. Yoni Bach is the associate.
Hello, everyone. Very nice to be here. Thank you for the invitation.
Yoni Bach is the Associate Vice President of Reagent and Consumable R&D at VizGen, a young Boston-based spatial transcriptomics company that uses Xiaowei Zhang's Murphish technology for their platform. Yoni has spent his career working in multiple areas of genomics. Before VizGen, Yoni spent time at The Broad, Illumina, 10x Genomics, and Celerity. We'll hear today about Murphish and where Yoni thinks spatial is headed. Hi, Yoni.
Oh, thanks for having me.
And of course, not all spatial biology is transcriptomics, and that's why we're thrilled to have Julia Kennedy-Darling joining us today from Akoya Biosciences, a company focused on spatial beyond the transcriptome. Julia, the head of Codex Research and Development at Akoya, was a co-inventor of the Codex technology while doing her postdoc in Gary Nolan's lab at Stanford. We look forward to hearing more about Codex today. Thank you for joining us today, Julia.
Thanks so much. Great to be here.
So whether analyzing genes or proteins, spatial biology or spatial omics goes beyond traditional single-cell genomics. In addition to informing which genes and proteins are expressed, spatial biology reveals where they're located. This spatial information is critically important when uncovering the biology of complex environments, such as a lung infected with SARS-CoV-2, or differentiating the complexities between a cancer tumor and its microenvironment. We're very excited to focus on spatial today, the technology, which was Nature Method's 2020 Method of the Year, is in its infancy, but is already creating a massive buzz in the genomics community and beyond. And we would like to answer your questions too. So please put any questions that you have for our guests in the Q&A section of the Zoom. Before we dive into each of our guests' individual backgrounds and the technologies that they developed or work with, we're going to get started with some Spatial 101. Kevin, I'll pass it over to you now.
Thanks, Josh. You've drawn the short straw. What is spatial biology? We've heard it called many things. Spatial omics, just spatial. Does the terminology matter? What is it?
Yeah, as the name sort of indicates, spatial biology is really understanding biology in its true native environment. instead of understanding the average response of large population of cells, to understand heterogeneity of each individual cells. But for example, knowing how many different types of cells are existing in the sample is obviously important, but how they are organized in space is actually incredibly powerful information. So, for example, if you have two biological samples with exactly same number of immune cells and the cancer cells, but the actual biology can be dramatically different based on how they are spatially organized. And this type of single cell or resolution is not the endpoint of the spatial biology. It goes much more than that. For example, we all know the function of the individual cell is determined by orchestration of multiple different types of biological molecules. So understanding the subcellular location of different biological molecules such as RNA, protein, or even structural elements within each individual cell is also part of the exciting promise of the spatial biology.
Yeah, great. Yoni, when did the first spatial technologies start to appear? Maybe you could give us a very, very brief history.
I mean, in reality, you can go back hundreds of years and go to a microscope and you have a spatial. So reality is a lot of the genomics kind of base that we're thinking of a decade or so, a little bit longer than that. Or if you really want to go back, where someone took an individual cell out with a pipette and put it into a tube and did some kind of PCR or qPCR reaction, going back even before my time at that aspect. So things have really evolved is the best way to look at it. This is a lot of different technologies that have been added upon each other over a century. We've had lots of new technologies, new enzymes that allow us to do new techniques, new imaging approaches. And I think one of the key things that is underappreciated is the ability to process all the data that we generate. So in addition to being able to take a picture, we can now process that picture and use the data successfully in order to do something that has biological relevance. It's not just a pretty picture, it's a bunch of data points. And as we go into more spatial omics, we're getting different types of data and we're getting spatial location. So instead of just counting, we can see where something is and count at that location.
Julia, we've heard some in the field compare spatial in terms of its potential scientific and maybe commercial impact to next-gen sequencing, which is a very high bar. Why is this advance so important to understanding biology?
Yeah, I mean, I think both Josh and Yanni spoke of sort of critical points of, it's really understanding the context of the tissue. And I think I like the analogy to next-gen sequencing of really sort of, I think next-gen sequencing opened up new frontiers because the ability of the technology sort of allowed you to ask those type of questions. So I think we're really kind of at the precipice of that point in spatial of, we're able to assess, tens to hundreds to thousands of markers within the spatial context. And now how can we apply that in different ways to be able to ask those type of questions? So I think, next-gen sequencing was a transformative technology
Sure.
and being able to sort of apply it to those type of questions and sort of revealing new questions overall. And I think that's exactly where we are at sort of this precipice moment in the spatial biology field.
Of course, next-gen sequencing arrived with some pretty expensive instruments. So Josh, I wanted to ask, how easy is it for researchers to enter this field and perform spatial experiments? Are the platforms and technologies something you would typically find in a core lab, or is outsourcing the way to go? Give us a sense for people who may be still new to the field.
Yeah, I believe the field is still young and emerging, and I think initially it was used by the scientific research community in various answering various different biological or questions, but the spatial biology, one of its promises and maybe the exciting potential is potentially applicable for healthcare. For example, if you look at the the current pathology, for example, microscopy is already part of the existing pathology workflow. And with the kind of resolution and sensitivity that the spatial technology can offer, because of the long-term aspiration and the direction of that this technology can go to, I think it's extremely important that the easy of use aspects of the technology, so that it is really widely accessible. For example, as you know, the microscopy is generally, especially the high resolution microscopy for molecular or imaging, is considered a field that requires a lot of expertise. But I think part of the challenges and opportunity for spatial biology is by making it very easy to use, to make it widely usable and accessible by non-experts.
Thanks. Yoni, what areas of research would you say have started to really see the benefit of this technology, and where do you see it having the biggest impact?
Yeah, I think if you look at things that are more immediate, it is more biological systems that single cell approaches are not applicable to. For instance, cells that are either too big, cells that are entangled with other cells, for instance, the brain. If you looked at, for instance, adipocytes, very large cells, but there's a lot of interesting, phenotypes that are involved in that process. So things that you can't use in a microfluidic system, or that are very hard to sort. And there's always the cells that are just difficult to isolate on their own. If you go through a microfluidic approach and you're dissociating your cells, you're actually losing large populations of different types of cells. And based on how you do your dissociation, you could lose different types of cells. So you kind of have to be selective. Where spatial is really, one of its real strengths is there's minimal, processing of that cell other than getting your sample ready for that imaging process.
Thank you so much. And finally, in this round, Julia, what would you say has been maybe the biggest success of spatial biology to date? I'm wondering if you could perhaps point to, is there a specific paper that's been published in the last couple of years, or just a discovery that you think really sets the bar for the field?
Yeah, no, that's a great question. I think maybe even the last couple of weeks. So Janice Taub and her colleagues published a paper in Science in the last few weeks, which really demonstrated sort of the capability of spatial biology to extract a clinically relevant subset of markers that can be associated with a clinical outcome or a clinically relevant outcome. And I think that's really the culmination of a lot of work and a lot of things coming together. So in that way, they were able to sort of say this signature of markers is indicative of being able to respond to certain immunotherapies, which are clearly sort of a very relevant application space for this type of technology. So I think that's one example, and there sort of are starting to be more examples of that type, where it's sort of, not just measuring your CD8 killer T cells, and not even sort of CD8 plus, say, your FOXP3 Tregs, but it's sort of the combination of those plus other markers, which really correlate to a clinical response. And that's, I think, the true utility of these type of technologies.
Fantastic. Well, I'm going to pass it over to Julianna, who is going to, we're going to probe a bit more about Murphish. Julianna.
Yeah, sure. Thanks, Kevin. So Yoni, can you tell us a little bit about your background and what made you want to jump into a young company working in spatial?
Well, yeah, so that's a very interesting question. I started off, I would say, going back, I was in pharma initially doing a lot of chemistry stuff. I did mass spec, NMR, and so forth. But after my time at the Broad, I actually jumped into genomics and went to Illumina and helped build a lot of the different sequencers. And seeing how that technology is being used and the different things that you could do back then, I mean, this was a decade ago still, and there's lots of really interesting things, and the scale of genomics has really increased. But really, what you're doing with genomics, you're counting a bunch of sequences that you get out of a library prep that could or could not be biased. From there, I went to 10x Genomics. I'm like, okay, what's the next big thing? What would be interesting next? I'm like, oh, we can isolate single cells. This field looks like it's evolving. I went there, and you look at the single cell data. You can trap a single cell into a microfluidic drop, and you can read out the RNA from there, and you get all this great information. But once again, if you look at the microfluidic-based approaches, you're limited to the cell types. You might not be able to read everything. There's the cell dissociation. The sample prep can be a challenge for the user, but you're still getting all this new information. And it was very clear, as you see a lot of the spatial stuff was coming out, for instance, Murphish, it's coming out and you're seeing all this data. You're getting massive amounts of transcripts. You're getting spatial data. And you're actually getting more unique reads than you would out of these other genomic approaches, because every time that you're getting a read, it's a single molecule. So you're reading a single molecule and you're getting 100 million reads in an experiment, looking at hundreds of thousands of cells rather than a few thousand cells at a time. I mean, I think for me, it's an obvious jump to a new type of technology there, to something that's giving you also spatial information. So you can tell that cell A is next to cell B and it has an interesting phenotype. Or if you're looking at an immune response, are those immune cells actually, attacking a tumor cell? Are they, attacking a cell that is infected with coronavirus, for instance? These are things that you can figure out from spatial technologies that you can't figure out from previous types of technologies that I've worked on in the past. So it was a clear decision, and startups are amazing. So I really encourage those.
Okay, terrific. And can you just give us a walkthrough about the nuts and bolts of Murphish? So how does it work?
Yeah, so Murphish is a targeted approach. It's based on single molecule fish approaches where we tile on different pieces of DNA that hybridize to an RNA. That those molecules each have a series of barcodes on them that are read out in a cyclical fashion. So you end up with a series of ones and zeros that you get from reading different signals in different cycles. You can then go to a code book that then reads out what that specific target was. The way that it's also designed, there's error-corrected options in it so that you are able to identify, if there are certain errors. If you look at a lot of genomic techniques, you'll notice you have to get rid of a lot of the noise first. This approach allows you to generate your data and also have an error-robust approach as well.
Okay, great. And how many transcripts the expression of how many transcripts can be read right now by the platform?
So the current MERSCOPE platform goes up to 500 genes very robustly. These are 500 custom genes that a customer could pick themselves. But if you look through the publication record, Murphish has gone up to over 10,000 genes at once. We're just building a method that's very robust and reliable right now, but we allow a user to customize the gene panels that they're going to look at.
Okay, got it. Okay, I'm sure we'll come back to more about Murphish in a bit, but right now I'll pass it over to you, Kevin.
Thanks, Juliana. You're watching GenLive, the first anniversary episode. We're talking about spatial biology, and we welcome your questions. They're already starting to come in. Please enter them in the Q&A box for our three guests. Julia, back to you. Tell us a little bit about Akoya, how it got started. I think it was in 2015, and your role in bringing Codex to the company.
Yeah, so you're right. So the company was started in 2015, and the idea was generated in Kary Mullis' lab. So there were sort of multiple very talented postdocs in his lab. So Yuri and Nikolai were two of those postdocs who came up with the very original idea, and they worked with Kary to start the company. So the idea of Codex is basically to tag antibodies with oligo tags, such that you can decouple the association of the dye to the antibody, and then sort of control when you read out the tag. So sort of similar to how Yanni was describing sort of barcode sequences that you hybridize to and then detect the signal. This is done on a protein level through antibodies that are tagged, and then we hybridize and dehybridize dye-labeled oligos to read out the protein signature. So the first generation of the technology was based on an enzyme-based approach where you'd read out the antibody signature based on that type of method. I got the opportunity to join Kary's lab and contribute to the technology and worked on sort of the next generation of technology, which was what I just described, which is the hybridization approach of sort of hybridizing imaging, dehybridizing. And then in 2017, I actually decided to transition over to Akoya at the time as the second employee, which is really, a great opportunity. We've grown really quickly over the last few years. So we had the opportunity to actually sort of join forces with the Fanoptics division out of PerkinElmer. And so, over the past few years, we've been able to combine the multispectral imaging of the Fanoptics portfolio with the Codex chemistry, where we really have sort of a continuum of technologies to meet different needs, depending on whether that's a discovery need or a clinical need. And I guess, we actually just had our IPO a few months ago, and, we really sort of see a lot of potential in the spatial biology area. And it's really around sort of the Codex technology plus the Fanoptics portfolio.
I'm sure there are many in our audience who know exactly what Fanoptics is, but I'm not one of them. Can you say a little bit about what that is and also what makes the Codex technology different from its erstwhile competitors?
Yeah, so I guess I'll answer your first question first. So Fanoptics captures a set of microscopes that use multispectral unmixing to be able to measure 6 to 8 parameters at a single time. So this is really well suited for, if you have a panel of, say, 6 different antibodies that might be critical to understanding the signature of a tumor microenvironment. And so this uses TSA dye deposition in combination with the multispectral imaging of the vector polaris, for example. Codex is suited for imaging something along the lines of sort of 10 plus proteins in a single sample. And so we use the same dye molecules per round and sort of add and remove them per cycle. And in that way, we can scale to be able to measure tens of protein markers in a single tissue.
Do you see Akoya potentially getting into transcriptomics, or are you going to stay in Fanoptics?
So that's a great question. I do have to be a little bit more careful with my answer now that we're a public company, but I will sort of tell you a few things. So I think at its core, what Codex is really sort of a modular system. So I think if you look at our last few press releases, we've announced our I² network, which is really being able to combine our FluidX instrument with other types of imaging platforms to really stay at the bleeding edge of this type of development. So in that way, it sort of demonstrates that our instrument is meant to be modular and to be able to control sort of the FluidX for different types of applications. And then at its core, Codex, from a chemistry perspective, is sort of cycling reagents. So it doesn't take a huge leap of faith to show that, or to think that you can sort of combine these ingredients in a way to detect something other than protein. And so I think, you can kind of put it together to show that this is something we might be interested in. We don't necessarily have a timeline associated with that. But I think in the end, we definitely see that, the spatial biology field is benefited. There's a lot of benefits to both sort of detecting protein and RNA. And ultimately, what we hope is sort of a multi-omics solution where you can put together multiple different ingredients to be able to measure the spatial context of lots of different moieties in the same sample.
Yeah, you have to be careful what you say now, you're public, but
yeah.
Maybe you can answer this question. Akoya recently stated that it will partner with AstraZeneca to advance some new multiplex immunofluorescence workflows and spatial biomarker signatures. How will your platform help AstraZeneca in drug development, do you think?
Yeah, so this is really around sort of our clinical aspirations. So I think, historically, when you analyzed a tissue specimen in the clinical realm, you might be looking at one marker or maybe 2. And so I think in order to, I think we sort of understand that clinically relevant signatures might be actually sort of up to 6 markers. And I think in order for that to be relevant in the clinical space, it really comes down to, do you have a robust technology and can it really scale to meet the needs of a clinical solution? And that's really sort of the Fanoptics arm of our technology, is using that methodology, both the TSA and opal deposition, plus our multispectral imaging platforms, to be able to robustly measure those signatures. And I guess the paper that I mentioned earlier with Janice Taub's team really sort of starts to demonstrate that seems viable. So I think that's a way that we would directly contribute to their clinical aspirations. But I think even before then, we sort of see that people start with discovery questions where they might want to understand what are the markers that are critical. And in that case, you do need technologies like what Yanni just spoke of with Murphish, of maybe 500 to 1,000 markers. Then you want to start to understand what those signatures might speak to. You then might want to ask, of the 30 or 40 in a smaller targeted panel, which ones then start to correlate. So I think we definitely see it as sort of a funnel where, depending on where you are in that continuum, you have different questions and different needs. But ultimately, it needs to result in a very robust panel that can be used directly to measure clinical samples.
Thank you. Thank you so much. I'm going to hand it back to Julianna.
Sure, thanks. So Josh, can you tell us a bit about your background and how you co-founded Rebus?
Yes, I started my college as a biology major, but I also wanted to, I got gradually moved towards engineering, and for my PhD, I was one of the developers of the new optical imaging technology at MIT, and so my background is primarily optical instrumentation, but I always wanted to apply this to the field of genomics. And when we co-founded the former version of the GenLive system in 2006, we wanted to apply the optics for doing a sequencing application, next-gen sequencing application. Around 2012, we also started applying the technology, same technology, to imaging cells and tissues, especially the single molecular or RNA fish assays. So that was kind of the starting point where we have made the optical platform as a chemistry agnostic platform in 2017, so that we have an opportunity to apply the optics for various different biological applications, including RNA, immunofluorescence, and DNA fish, and various different applications. But as you know, there has been really dramatic progress in microscopy-based detection of various biological molecules, chemistry methodologies, and we have launched recently the spatial omics platform called Esper, and we have just started shipping the boxes. So quite excited about where we are.
Okay, great, thanks. So yeah, a lot of spatial technologies require some form of sequencing, but Esper, I think, is all imaging based. Is that correct?
Yes.
Sequencing needed, right?
Yes.
So can you tell us a little bit more about how that works?
Yeah, first of all, sort of the underlying motivation for us to develop this platform is that we believe that in order to fully realize the full potential of the spatial, promise of the spatial biology, there are important needs that are not completely mapped by the conventional high resolution microscopy because of how it works. It's sort of a fundamental limitation imposed by how the lens works. For example, if you want to have a high resolution that detects individual molecules within the cell and its spatial location and how it's related to the other biological molecules, you do need a high resolution microscopy such as 60x or 100x foil, but you have to take the very dramatic, for example, throughput and easy of use, and also the range of sample format that you can apply the high resolution microscopy to. For example, if your tissue is on top of a 1 millimeter thick microscope slide, then there are types of high resolution imaging that you cannot apply. So this is actually a well-known microscopy, and a lot of biologists take it for granted. If I want to have a high resolution to image molecules, I have to take sacrifice in the number of cells that I can image, and also I have to do something called Z-stack, which slows down the imaging operation and so on. So this is sort of a what we wanted to do is there any way that we can sort of decover so that, so that we can continue to improve the resolution without sacrificing the field of view and throughput and ease of use. And so this is what we thought that what we thought the opportunity was, that there is indeed a sort of imaging bottleneck in single cell biology. And we wanted to address that problem using synthetic aperture or optics. And the way it works is actually sort of similar to well-known microscopy technique, or structural illumination microscopy. Our approach is a little different from the conventional structural illumination. We deliver to the sample plane high-resolution light pattern that are formed by the interference of the propagating laser beams. Each of the laser beams are the plane waves. It's not a focused laser beams. That gives us an opportunity to engineer a light pattern and design the light pattern and use it in a way that really you can really boost the resolution much beyond what the lens and the camera is capable of. So we're currently using 20x low numerical aperture lens with a numerical aperture of 0.45 that offers enormous advantage in field of view and depth of field and the working distance, but use our structural illumination technique to drive the resolution close to 100x oil or XY resolution. So we believe this offers a way to execute multiple different chemistry methodology that are designed to specifically label not just the mRNA, but also protein and even structural or morphology information. We hope that the high resolution, sort of the combination of the high resolution and the large scale, is something that we want to deliver to the scientific community.
Okay, terrific, thanks. We have a lot of great questions coming in from the audience, and I know Alex has some questions as well. But before I pass it over to you, Alex, one quick last question for you, Josh. We heard that at Rebus, your naming scheme is very meaningful. And can you tell us why your platform is named Esper?
Oh, that's a very hard question. That was done by our brilliant marketing team.
It's very specific, I know.
Yeah.
I heard it had something to do with like hope and like esperar, like to hope. So hoping.
That's right. I do know that it is related to the hope, and we also really aiming to deliver the device that are really easy to use, that are friendly. We want the spatial or omics to be an experience where really democratized. It doesn't really require the expertise. So that's kind of we wanted to come up with a name that appears to be very friendly, but also reliable.
Okay, great. All right, I think Alex, I'm going to pass it over to you for some more questions.
Sure, thanks, Juliana, and to all of our guests. I'd like to get your take on where your companies are, or where do you perceive yourselves in the progression and evolution in the field of spatial biology?
Yanni, do you want to go first? I'm happy to jump in.
Sure, go right ahead.
And then I'll jump in after you.
Sure. Yeah, so I think, we, Akoya, we call ourselves the spatial biology company. So I think this is really sort of our whole focus is spatial biology applications. And I spoke earlier how we have solutions for protein relevant detection today. So I think, from the discovery realm through the clinical realm, with the middle sort of representing the translational realm. So I think really where I see ourselves is we have really robust, strong solutions to meet the needs within that space of sort of for protein detection from, say, 50 plus markers through a very sort of robust panel where you're more inclined to sort of focus on throughput. I think that's really sort of our strong suit right now. And I think where we're headed is, we think of the Codex technology specifically as modular and sort of being able to conform to sort of what's next. And I spoke of, ultimately we see that a multi-omics solution will likely be helpful, or as sort of be able to answer the questions that people might have. And so I think where we're headed is, wanting to be able to answer those multi-omic questions directly with a combination of our protein technologies and maybe either partnerships with other companies who can provide some of the RNA solutions, or even sort of developing solutions in-house to be able to meet those needs. But it's really sort of around the protein side of the street today.
Yoshi? Yoni?
So, from our perspective, right, we're looking for a system to build for customers that they can go from sample to answer and not be dependent on other systems as well. If you look at a lot of the technologies that have been developed in the past, you have to sequence something or you have to buy another system from someone else. Our system is sample to answer, a way to prepare your samples, a way to image, get your transcriptomic data, and also be able to process it as well. So we're kind of pulling people out of that kind of being tied to multiple companies, and you have to spend $1,000 here, $1,000 there, and then you end up paying way more than you thought per sample. With regards to kind of the future direction of the technology, other than making it simple and making Murphish accessible, because if you look at the technology, there's a lot of different things you have to understand. There's imaging, there's the chemistry, there's the microfluidics, there's the computational part. We provide the answers for all of these different issues that someone has to handle when running a Murphish experiment, for instance. And then technology in the future, obviously, we'll expand on how many genes someone could target, for instance. There's some other kind of obvious things you might be able to find in literature that might be integrated, but our exact pipeline, we're still a small startup, right? Limited release. There's a limit to how much I can actually discuss with you with regards to our pipeline, but anything that you could target with a hybridized piece of DNA, in theory, you could measure that on the Murphish system. So that's the best way to look at it.
Josh, any thoughts on one or other?
Yeah, we, as I mentioned earlier, we just launched the automated platform earlier this year, and we designed this to be, strong emphasis on automation, as you already mentioned, which I think is easy to use, is important. We designed the instrument that in addition to the resolution and high sensitivity as a, in terms of the scale, we currently offer about 3 centimeters square, or imaging area, which is very easily further scalable. So we think that this will accommodate use of multiple tissue sections that are run on the same single instruments. And with the end-to-end integration, we have tried to make it fully automated. So the total hands-on time from the tissue section on the researcher's hand to the spatially annotated cell by gene matrix is really less than 1 hour. So we would like to provide such level of automation to the whole process. We have started applying our optics to a single molecular or mRNA fish field as early as 2012. And the kinds of chemistry that we have just launched is sort of allows very direct visualization and quantification of the gene expression simply by counting. So it offers the absolute quantification over large dynamic range. So this is the kinds of chemistry, in addition to the high sensitivity, but applicable beyond the cell types analysis, so a lot of disease markers and the cell status markers require ability to measure gene expression with absolute quantification. So beyond this, we definitely are aligned with other speakers. Emphasis on multi-omics aspects here. So we are working on adding protein in addition to the RNA, but also applying our optics advantage to additional tissue types such as the FFP tissue types. So, so that's roughly the direction that we are going. We're very excited to continue to update the progress to the market.
Great. You mentioned the progress now. What do each of your companies see as your end game, so to speak? For, I know Akoya just went public, and for Vizgene and Rebus, is it staying independent, someday combining with some larger entity, or where do you see the companies down the road?
I'll let you go ahead.
Yeah, we are relatively early stage company, but Rebus Biosystem as a company made an important transition this year to by launching our product. We are currently building quite rapidly our sales and the marketing team and the commercial operations. So it's very excited to be at this stage, at this market. We want to continue to build our manufacturing and ability to continue innovate. We think that this is a early market where we want to be very careful about how to prioritize things and so on. So we're relatively early stage, but we're very focused on achieving commercial success of our initial version of the product. But we also are very looking forward to adding additional chemistry kits. One of the things that we think is important is the flexibility of the platform, so that the researchers can more than one chemistry to answer a wide range of biological questions. So that's another strategy that we are taking here. So we're very looking forward to launching another assay kit that can provide more than thousands of the genes as a complementary assay to the existing assay that are in the market right now. So we have a lot of exciting goals that we are pursuing. So, but relatively we're early stage at this point.
Great. Julia or Yoni?
Yeah, I mean, I'm an R&D person. I don't do that kind of buying and selling of companies. But I mean, we did get our Series B recently. We're rapidly growing. We're a small company, and we expect to double in size this year and grow a lot next year as well. So we're kind of on that trajectory. I mean, I can give you like examples. Illumina wasn't a sequencing company in the beginning. It was a merger of several companies. So in the long run, you don't know what's going to come out of the companies. There might be right fits where companies merge and create some new phenomenal technology. There can be situations where you go the IPO road. I mean, that's the best I can answer that question.
Mm-hmm. Julia?
Yeah, I think I'll also play the R&D card that these aren't the talking points I typically take on, but I think, I think we obviously just hit a big milestone with our IPO. And I think where we see ourselves, I think a bit more in terms of functionality and sort of the applications that we're targeting. So I guess said this a few times, but I think it's that we see that there's a lot of relevance clinically in these type of approaches. And so I think ultimately that's what we want to deliver on. So I think that's really what we're focused on at the moment.
You're on GenLive. We're talking spatial biology, and just now talking about capital, and why has spatial biology taken off to the point where companies are growing and attracting capital, whether that's privately or publicly, as in the case of Akoya?
I guess, so I think one reason is, sort of concurrent with spatial biology, and I don't think they're unrelated. You know, cancer immunotherapy has also had a lot of attention in the last 10 years. And I think the two really go hand in hand. And I think one of the reasons that spatial biology has really come to the forefront is, especially within cancer immunotherapy applications, you typically can't answer why certain patient populations respond to certain therapies when you're looking at just a single parameter, or even just a single parameter in the spatial context. So I think this is really where there's a need to be able to have these type of technologies to say, you know, it's the dendritic cell plus the granulocyte plus the cancer-associated fibroblasts, and where they are spatially, that gives you that correlation. And so I definitely think it's because of the interest in the cancer immunotherapy realm that has really driven a lot of the innovation in the spatial biology area. There are certainly other applications that benefit from this type of technology, but I think that's really the impetus.
And what sort of customers are most drawn to your solutions or platforms? Among the company, how are they alike or maybe different from company to company?
I mean, maybe I'll jump in there a little bit. I mean, it's a diverse set of customers that want to look at different types of data. There's people that have looked at a data type and said, there's a limitation that I can't address with what I have. They may be able to do some kind of simple imaging. They might be able to do single cell or just genomics in general. But people are looking for a solution that they can look at a piece of tissue, they can read out certain piece of information, know which cell is next to which cell, know what transcripts, you know, or proteins or whatever they want to target, and actually see that and get data that is actually almost all of it is useful. You don't need to filter through a bunch of, you know, ribosomal RNA or some, mitochondrial RNA that isn't particularly helpful. You get good data that's really relevant to what they're looking for, and it can be very targeted and very specific.
Josh or Julia?
So let me jump in here. Yes. Well, we see a lot of researchers. Most immediate, maybe obvious need is that a lot of groups who has done single-cell RNA sequencing, they want to validate the result. They have the set of markers that they want to detect in their tissue sample with the information about how those are spatially organized. Another example where the researchers find our platform to be really powerful is the ability to measure or expression level and be able to link it to the specific cell type or anatomical structure. For example, there was one case where there was a complex disease such as Alzheimer's disease, where we can look at the tumor and the disease tissue in one flow cell experiments and be able to really detect subtle but incredibly important difference between those tissue types. This is a kinds of information that will not be accessible by existing technique, because you need the ability to do very precise quantification of a gene expression based on microscope-based direct quantification, but be able to link it to what is the specific, how is that different from specific cell type and specific anatomical structure. These are the kinds of resolution and the information that are just becoming available based on the spatial technology. So we also have a lot of applications that we have been applying for the cancer research area, where these fields tends to have relatively well-defined biomarkers that are 30 to 40, 50. And they're just looking for a very simple, easy to use and reliable device to reliably detect these markers with high resolution and sensitivity. So these are the areas that we have been really having exciting success recently.
Great, and we're still taking audience questions. If you have them, you can use our Q&A box to ask them. I believe
let's
Kevin,
go into those. Let's go into those now, Alex.
Sure.
Kyle asks a couple of good questions, actually.
Yeah.
Can you talk about the progression of spatial biology technologies from research and biopharma settings into the clinic, obviously diagnostics and so on? How long could this take, and which technologies are furthest along the path to the clinic? Julia, maybe I'll start with you.
Sure. Yes, I think sort of the one differentiator, sort of what are you measuring? So I think in terms of what's farthest along, I think, protein targets tend to be the furthest along because those tend to be what's actually targeted in a lot of the drug therapies. And then I think, I spoke a little bit about the considerations are different when you're thinking about a clinical solution versus something in the discovery realm. So I think it's really around throughput and robustness and being able to really accurately measure the specific markers that you're going after. So I think something along the lines of, one of the, something within the Fanoptics portfolio, so some of the Opal panels that are measuring, say, 6 markers, that's currently, I think, the closest to this type of solution. In terms of how long it would take to get there, I hate to speculate because I don't have a specific answer, but I think, it does take time to be able to put together sort of all of the information you need in all of the studies to prove that viability. But it is interesting, sort of, you do have very different considerations when something clinical is your eventual goal.
Yoni or Josh, you want to add anything to that?
Sure, a little bit and our system is definitely an RUO system now, but you have to appreciate, like for genomics in general, most things that ended up in the clinic, at least early on, those are lab developed tests. So what someone would take an RUO system and then validate it in a specific lab that is doing clinical research. So the shortest path for a lot of new technologies, that's how it's going to end up in the clinic faster. The validation to go into some kind of, let's say, a CLIA or IVD type experiment, it takes a lot of time and dedication. And when you're building up a technology, you don't always have the resources to do that. But then partnering with the right, labs, you can build out these lab developed tests and get to that clinical stage earlier than you would have expected.
Okay. We have a Murphish question. How feasible is it to do Murphish in 3D cultures?
Oh, geez. Well, right now we do go a little bit into the Z stack, but I wouldn't put it as particularly like a giant 3D system. Right now, our system is not a 3D system. It's kind of a 2D 1 centimeter by 1 centimeter imaging area, and it goes about 10 microns in depth. But we do Z stack through that. You could, go a little bit deeper, have more Z stacks, but there are advantages and disadvantages of going into a Z stack. There's more imaging time. So right now, I would say that's more of a not our thing at the moment, but there is the ability to do, 3D with most optical type systems eventually.
I'll jump in with another question, Yoni, for you, and it's basically linking how you get the spatial information from the sequencing data. So the question is the spatial information derived from the sequencing data itself, and how are imaging and sequencing data organized or put together in the studies?
Well, our stuff is based off, we're targeted in our system. So you know what you're targeting, all right? So we have, we hybridize along a piece of RNA, and we have different barcodes hybridized onto it, and we can image that way. From a sequencing-based approach, and we don't do sequencing. We are imaging based off of hybridizing DNA to RNA. Other approaches, sequencing-based approaches, I don't really work on those right now. So I'd probably let someone else comment on the sequencing-based imaging approach.
I actually think none of our platforms are sequencing based.
Yeah. Okay, well, actually, there's another Codex question, so we can move into that one. So in Codex, using Codex, is there a need to wash the sample for every sample imaged?
Yeah, so there are a series of steps up front of imaging, and then once you enter that part of the process, each cycle is basically comprised of 3 steps. So there's hybridizing, imaging, and dehybridizing. In a sense, what you're doing is washing the sample both during the hybridization and dehybridization steps. So those are both isothermal, but there are sort of built-in washing steps as part of that process.
I have another question from Kyle. Are customers looking for an end-to-end spatial platform that can address questions across the complete spectrum of research, discovery, translational, and clinical trials? For example, Julia, is this something Akoya is looking into? In other words, an instrument that combines features of Codex and Fanoptics?
Yeah, so I definitely think that customers are looking for that. And there are multiple reasons, but one of them is in any sort of high-plex technology, there's some time it takes to optimize the moiety. So whether it's an RNA moiety or a protein moiety. And I think the idea that you'd have to sort of switch platforms once you either sort of increase or decrease the panel and re-optimize, that's just not a desirable end state for the customer. So I think sort of that type of modular approach where you could use the same platform, the same instruments, the same reagents, and tailor it to your needs. So whether it's sort of high throughput, low parameter, high parameter, lower throughput, but sort of more data per sample. I think there is a desire to have such a modular system and not have to re-optimize the setup each time.
Okay, thank you. Alex, pick a question.
All righty. Yeah, I saw one over here from Colin. What is the biggest hurdle you face in having new customers adopt your platforms, to any or all three of you?
I can take a
Sure.
Go for it, Yanni.
Sure, Julia, go right ahead.
Sure. So I guess I'll give one perspective and then Josh and Yanni, feel free to jump in. But I think it sometimes depends on the perspective of the customer. So I think this speaks at one of the previous questions, which was who tends to be your customer base. And I think we have customers who might be a pathologist who are sort of very well versed in looking at one or two parameters, and then they're looking to sort of expand to say 20 or 30. I think their lens that they're viewing the technology through is quite different than someone who's coming from, say, a non-spatial technology, but something that's very high parameter. So, 50 parameters isn't going to scare them, but then how to analyze it in space is sort of the key question that they need to understand. So I think we've definitely seen this dichotomy depending on sort of where they come from and what their previous perspective or training is. There's sort of different aspects of the technology that we like to speak to make sure that they see the value where sort of they're not scared by some aspect of the technology. But Yanni and Josh, I'd actually be curious to get if you sort of see this similar thing with your customers.
I find that there's a lot of desire for spatial technologies right now. Right? It's more about, how do I get some data into a customer's hands so they can analyze it and see what benefits that they're going to get out of it? How do we get enough, how can we generate it for them? How do we work with people? We have a lab services arm that, people can generate data with their own tissues and see what they're getting. You get a lot of really great, fantastic data, when you generate these systems. And at that point, it's just getting the systems into the customer's hands. I mean, that's the biggest challenge. And honestly, right now, plastics are hard to get.
Josh?
Yeah, we're doing similar, or we're working with various different researchers for technology access program and lab service programs. So we're increasing the number of different types of biological questions that our platform can be applied to. And this gives us a really wonderful opportunity to understand the market's need. We have just recently started shipping our instruments, so we are very much looking forward to understanding the direct feedback from the end users as well.
Haley has a quick question. Apologizes for being late. Is spatial genomics more compatible with long read or short read sequencing?
Well, we don't do sequencing. I don't think any of our platforms are actually
Okay.
sequencing based, right? So there's kind of, that we're all imaging based. And so we don't require sequencing, which I think is a big advantage. We're not tied to a different type of technology and have to work around that. We each have our own technology that is applicable within our systems.
Right. Well, we apologize to Haley for not having the right panel to answer that question. Juliana.
I do know that some of the sequencing base are run on Illumina, so that could be at least step in the right direction for that one. Well, I think, oh, the last thing I wanted to mention before we wrap up is that Sandra had a question, are there any pictures to illustrate the different technology? And I was going to say, each of these companies' websites have beautiful pictures. I mean, one of the really fun things about Spatial is the amazing images that come out of them. And also on our Gen website, as we write more and more about Spatial, we also have a lot of pictures up as well. So you can definitely check those out. And that brings us to the end of this session of GenLive. I want to again thank Julia, Yoni, and Josh for a terrific discussion. And we at Gen look forward to following this field in the future very closely. And I want to thank Kevin and Alex and the rest of the Gen production team for making this episode run so smoothly. And thanks again to all of you for tuning in. Don't miss our next GenLive in July, where we'll be looking at the latest advances in agricultural biotech and genetic engineering. I'm Julianna Lemire. Stay safe, and we'll see you next month.